Hello, biology students. This is Mr. Russell here at St. Charles High. I hope you're having a wonderful day. I'm back with the third installment of the notes for protein synthesis. That's our 7th objective, the first one here in the spring semester. Okay, we've already talked about proteins and enzymes and how they're structured, and also how they function a little bit, and then we talked about transcription, how we copy the information in DNA into mRNA so that we can use that to copy, well, not really copy, we use that information to make proteins out of it. Okay, well, here, this third installment is a little bit shorter. We don't have to cover quite as much info. We're really just talking about the process of translation, which is how we take that mRNA, as you can see here, and actually use that to make a protein. This occurs in the ribosome. You may remember from last semester, Objective 2, that ribosomes are the site of protein synthesis in cells, and that's what we're going to talk about a little more today. Okay, another part of this overview, mRNA, we talked about how there's 3 base sequences on it called codons, and they're going to match up with the anticodons, which are complementary on tRNA. And doing that, we're going to add amino acids together using peptide bonds, and eventually we're going to have a whole protein. Okay, so let's get started. All right, so the basic steps are, first, you start off with initiation. These also are the same names as the steps in transcription from the last objective. And you'll notice that they have the same names, but the things happening them are kind of similar, but they're not exactly the same, because instead of copying mRNA, we're making a protein. We do start out with initiation. Okay, and that's where the ribosome is actually going to bind. The 2 halves of it are going to clamp down on the start codon on the mRNA. Okay, then is the process where we actually build the protein. This is called elongation because it's a long chain of amino acids and we're making it longer. All right. Anytime you see AAs, that refers to amino acids. By the way, you'll see that shorthand here and there in this presentation. Okay, so the amino acids are added to this growing polypeptide chain. That's another name for a protein. Remember that peptide is a name for amino acids. Poly means many, so it's something that's made out of many amino acids. So this is where we start adding amino acids to our chain in order to actually make a protein. This happens very, very quickly, about 15 or 20 amino acids per second. And you end up with a few mistakes along the way. About one mistake about every 10,000 nucleotides. Okay, so that sounds really rare, but considering how often this is happening on all your cells, it happens pretty frequently. Fortunately, your cells are pretty good at fixing these errors. Okay, the last step is termination, and this is where we hit a stop codon, which is any of these 3 codons, UAA, UAG, UGA. And then when the ribosome gets there, it sees that, and it's kind of like the period at the end of a sentence. It knows that it's done. It breaks away and leaves to go start building another protein. Okay, this is kind of how it looks. You'll see a lot of different models in here, all representing the same thing. These strange little plus sign shaped things are tRNA, and they are carrying amino acids to be matched up with the mRNA. So we use the mRNA to tell us which order these amino acids go in. Okay, so in a little more detail, first thing that happens is your ribosome is going to bind to a start codon. It's always going to find this codon to start with, because remember, just like school always starts in August, the mRNA always starts with AUG. Okay, there are 3 sites within a ribosome, and they're called A, P, and E. That's an attachment site. And then P is for peptide bonds, because that's where you actually form a peptide bond to attach a new amino acid. Then E is ejection. That's where we get rid of the tRNA that we've already used. Okay, so you can see here's a ribosome. You'll see you have the large subunit on top, the small subunit on the bottom. Our mRNA is kind of stuck there in between. And then we have 3 sites. You can only see, well, they do have the letters here, A, P, and E. Our tRNA comes in, attaches here, and then these things aren't going to move, but the whole ribosome is going to slide to our right. So it's going to, so this orange one started out where the purple one is, slides over one, It's going to attach its amino acid to the growing chain, so we're going to have a bond show up here in just a moment. And then afterwards, this bond that attaches the amino acid to the tRNA is going to break, and it's going to get ejected. Kind of like in the movies when you see a shell casing get ejected from a gun after it fires. It kind of happens the same way. The tRNA are ejected after they're used. Okay. mRNA codons match the complementary tRNA codons. Remember that anti, that prefix has a lot of different meanings, but here we're always going to use it to mean opposite, and they're opposite because they're opposite bases that attach to one another. Okay, at the end, it's going to release the amino acid from the tRNA, and it's going to join to the other amino acids so that you're actually building a protein here. I'll have a little animation in a couple slides that shows you how this happens. It makes a lot more sense when you see it in motion. Okay. Here's another diagram of the same process. You'll notice we have our mRNA in red and then our tRNA here in blue. The anticodon is also in blue. You'll see the mRNA is AUG, the start codon. And then a tRNA with UAC, which is complementary, so they match. If this were a CGG, it wouldn't fit here and it couldn't land there. So that's how they get things lined up correctly, is by matching these. Okay? So after it binds there, it's going to bring in its amino acid, which happens to be methionine, and then it's going to slide over to this spot, in which case another one will show up and land here. Another tRNA will land there. It'll bring its amino acid. Then the ribosome slides to the right again. Those two amino acids would be joined. And then it'll slide over again. And this tRNA, which no longer has an amino acid attached to it, is going to be ejected. It's going to leave, probably just go find another amino acid to bring back. Okay, I have an animation that shows you how that works right here. Okay, this is a translation. Hopefully you can see from the shape and the structure of this thing what it is. You can see it's a single strand, and you'll notice these are all A's, U's, C's, and G's, which means that this has got to be a mRNA molecule. It's not DNA, or else it would be two-stranded and have T's instead of U's. Okay, so let's start the process. Initiation, our small ribosomal subunit shows up and binds. Our first tRNA is going to show up, so the tRNA are the red. The amino acid is the little shape that's attached to it. I don't think they'll always be green. And then you'll see three bases on the bottom of the tRNA. That's our anticodon, and they have to match with the mRNA. Let me resize this. Okay, so as I was saying, the tRNA anticodon has to match with the mRNA codon. UAC matches with AUG. This mRNA is AAG, so what would go with that? UUC. Next one's UUU, so that would be AAA. So you'll have one of these red tRNA with those anticodons to match those spots. Okay, you'll also notice our large subunit of the ribosome is showing up to land here. Okay, it's going to attach. All right. You'll notice you'll have your three sites here, two attachment sites. The first one is A for attachment, because that's where they first attach. P is peptide, because that's where the peptide bond forms. And then E is for eject, because that's where this tRNA will leave. All right. Okay, so at the P site, we formed a peptide bond to attach this methionine and this lysine. And then it slides over again. So the tRNA that was at the A site is now at the P site, and the one that was at the P is now at the E. It gets ejected. Okay, another tRNA is going to show up here. AAA matches that. That has phenylalanine attached to it. We're going to slide over to the P site and attach those. So now we have three amino acids together, so we're starting to build a protein there. Once this one is in the E site, it gets ejected, and this is just a process that keeps continuing. New tRNA comes in, we slide over, we attach with a peptide bond, and then we get rid of that amino acid. Or not the amino acid, the tRNA, sorry. Okay, let's finish this out. Okay, it's called the restriction factor, and this tells it where to stop translating at. So this tells the ribosome, oh, we're done. It's like the period at the end of a sentence. That's a stop codon. You'll see that's UAA. The other ones are UGA and UAG. So if the ribosome sees any of those three, it knows, oh, it's time to stop because this other protein is going to come in and lock on that spot. Okay, and then everything breaks loose. Now this is not long enough to be a real protein, but this is how it happens. You'd have dozens or hundreds of amino acids making that one protein. Okay, oh, sorry, I said this was a restriction factor. It's a release factor. Sorry, because it tells everything to release from the mRNA. So it's done. So now this ribosome would float off and find a new mRNA molecule and start making a different protein. That process happens over and over and over again. Okay, let's get rid of this animation and go back to our PowerPoint. Okay, talked about that already. All right, last thing we really need to talk about, or the last step, is protein folding, which we've discussed already. I believe you've seen this before. Okay, so once we have this long chain of amino acids, which is what you see in gray here, because some of them have positive charges and some have negative charges and some don't have charges, that's going to cause some of them to start sticking together and then some of them won't. And you end up with a very complex shape, but that shape is very specific and it has to be that way. If not, then your protein may not function correctly, and that could be really bad. You could be born with some kind of a defect. It could give you a genetic disorder, or a lot of the times it may be bad enough that you may just never be born. You may die before you ever get even close. Okay, so that's how the protein folding works. And remember that even a little change in this shape based on a difference of amino acid sequence is going to change the shape of the protein and therefore could change its function. It may not always. If right here where the red is the attachment site, or the active site where it actually binds to the substrate and works, well, let's say we have a mutation over here, I don't know, that happens, that may not affect this protein's function. Okay? You may also have a mutation that happens right here and it actually helps the protein, makes it work better. Okay, so then this organism with these proteins will actually be better suited to survive. Okay, so a little summary to finish. Transcription and translation. You have your DNA in the nucleus. That gets transcribed into mRNA. So we add those complementary bases to make that single strand of mRNA. And then it's going to leave the nucleus and go out here. A ribosome is going to find it. And the ribosome is going to use these tRNA molecules to match the appropriate amino acid to the right spot on the mRNA. So this mRNA tells the ribosome which order to put the amino acids in. And then we end up with a string of amino acids, and that is our protein. Okay, I'm going to throw a couple links that you can click on this video, and they will take you to other resources that might be helpful. This is kind of a complicated process that confuses a lot of people, but that's how your cells make proteins. We'll work with it a little more in class. I hope this helps. If you have any questions, put them in the comment section or email me or write it down so you can ask in class. Thanks.